Okay, ladies and gentlemen, so let's quickly review the section 5.2, which is basically mitosis and interphase. So the first thing I'm going to do is I'm going to review some of the concepts that we discussed in class today. And we're going to talk a little bit about chromosomes, and we're also going to talk about interphase, and we're also going to talk about mitosis, okay? Okay, so the first thing that we want to go through, chromosomes. It says chromosome is one long continuous thread of DNA that consists of numerous genes along with regulatory information. So basically, a chromosome is a long strand of DNA that, if it were to be uncoiled, it would be about 10 feet long. And our body has about 46 chromosomes or 23 pairs. And the chromosomes contain a lot of genes with information and regulatory information, like to avoid mutations and other or to trigger other processes. It says that during interphase, or when DNA cells not dividing, DNA is loosely organized. And it says it looks a bit like spaghetti. So when the cell's not dividing, that DNA is not organized. Um, and or it's not that it's not organized, it's just that it's loosely organized and it looks like spaghetti. It's when the cell begins to divide, when mitosis begins to occur, which we're going to talk about in a minute, then those DNA starts to coil up inside. And this is important for that to happen, because if it didn't happen, that means that the genetic information would be all over the place, and that could cause a lot of mutations or a lot of other complications. It says during mitosis, the duplicated chromosome must condensed to be divided into two nuclei. And again, this is important because if they didn't divide into two nuclei, then the genetic information would not be correctly stored, and that could lead to mutations and other, like I mentioned already, mutations in other situations within the cell, or that there could be an error in the genetic information. It then says that DNA wraps around histones at regular intervals and gives structural support to the chromosomes so DNA molecules can fit into the cell nucleus. So basically, the DNA wraps around the histones, and the histones are very simple proteins, I guess structurally simple when you compare it to others. And what the histones do is it provides support and or structural support to a chromosome. And what it does is it allows the DNA molecules to fit inside the nucleus. So it allows the cell, the chromosomes, to begin to be binded, begins to help the chromosomes bind together and then condense. Because the loose combinations of DNA proteins are called chromatin. One half of a duplicated chromosome is also called a chromatid. It says some chromatids are held together at the centromeres. Okay, so the centromeres are the structures that hold the chromatids together. And the chromatid is basically one half of a duplicated chromosome. It says telomeres prevent the ends of chromosomes from attaching to one another, and they prevent the loss of genes. Now, the telomeres, genes are always lost, and they're lost because it's part of the DNA cycle. It's part of the reproduction, and it's lost because that's probably DNA that was damaged. And the telomeres help avoid other DNA, other important strands of DNA, not to be lost, okay? Especially those that contain very important information. Now, before we go to mitosis and we talk about the steps in mitosis, let's talk real quick about interphase. So when we talk about interphase, and I'm going to type it down here real quick, interphase is defined as the I'm sorry, as it plays an important role in preparing the cell to divide. Okay, interphase is where, let me see if I can, is or plays an important role in cell important role in the um in preparing the cell to divide. Okay, it plays an important role in allowing the cell to divide. And what it does is it goes on to provide critical time for the duplication of organelles and for DNA replication. So this interphase is very important because it's basically what we were discussing in the previous section. That G1 where the organelles uh, increase in numbers when the DNA or that S phase where the DNA inside the nucleus is being divided, and that G2 phase, which as well as the G1 phase, also provides important checkpoints for the DNA to or for the cell to remove any possible damaged DNA. So interphase is that very important plays that important role in preparing the cell to divide. Now, when we go to mitosis, we know that mitosis divides the cell nucleus into two genetically identical nuclei with its full set of DNA and is divided into four steps. So we're going to summarize the four steps that involve mitosis. And again, mitosis is when all the content inside of the nucleus divides. And the first step of mitosis is the prophase. And in the prophase, it says that chromatins condense into tightly coiled chromosomes. So in prophase is where the chromatin begins to condense into the chromosomes. In the second phase, which is the metaphase, it says that the spindle fibers attach to a protein structure and align chromosomes along the cell's equator. So basically what's happening in metaphase is that those spindle fibers that are attaching to the cells are actually pulling those chromatins into the cell's equator, which is the center of the cells. In anaphase, it says site chromatids separate from each other from the spindled fibers to shorten, pulling them to opposite sides of the cell. So in metaphase, basically the spindle fibers attach to the protein and align them along the center. In anaphase, the spindle fibers begin to basically detach or pull away, and what that causes is it causes the chromosomes um, to go to the opposite sides of the cell. And then in telophase, it says a complete set of identical chromosomes is positioned at each pole of the cell. It says that the spindle fiber disappears and the nuclear membrane begins to form. So it's actually in that phase, in the um, telophase phase, where the spindle fibers disappear, where the cells completely duplicated, or when the cell's chromosomes are duplicated. Still in the anaphase, chromosomes are not really formed yet, but they do begin to pull towards the opposite side of the cell. And it's actually in the telophase when that spindle fiber begins to disappear, and the cells are, the spaghettis completely disappear as well. That spaghetti DNA is no longer there, and that the chromosome is fully divided. And after that, after the cell undergoes mitosis, the last step in cell division is actually the cytokinesis. And real quick, I'm going to define what the cytokinesis is, and then that would be the end of this video lesson. Hopefully, this very brief summary will help you guys understand the process of mitosis and cytokinesis better. So, cytokinesis can be defined as the process that divides the cell's cytoplasm and into two cells and completes a full stage of the cell cycle. So, cytokinesis divides the cytoplasm into two cells and completes a full stage of the cell cycle. Let's see if I can move that. I think I can move that out here. All right. And completes a full stage of the cell cycle. All right, guys. So again, cytokinesis is the divides the cytoplasm into two cells and completes a full stage of the cell cycle. Okay, so this happens hundreds of times, and it happens in all of the cells in our body. So this is a very, 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 very frequent process. All right. So hopefully this video will help you guys understand this process. Again, prophase and mitosis, chromatids are condensed into tightly coiled chromosomes. In metaphase, the spindle fibers attach to protein structures and align the chromosomes along the cell's equator. In anaphase, is when the site chromatids separate from each other. Then the spindle fibers begin to shorten, pulling the cells to opposite sides, or pulling them to opposite sides of the cell. Excuse me. Telophase is a complete set of identical chromosomes is positioned, and each pole of the cell, spindle fibers disappear and the nuclear membrane begins to form. And in interphase, guys, is what we talked about, and in cytokinesis is when the cell's cytoplasm is divided into two and a full stage of the cell cycle has been completed. Hopefully this video will help you guys review those steps and the concepts better. So stay safe and we'll see each other soon. Bye.